Kate Scow here again, and it is a lovely Saturday evening. Well, the sun is setting, and this is Soil Microbiology, and I'm giving a video cast of the second part of Lecture 6, which is part of the nitrogen cycle, focusing specifically on nitrogen fixation. And this complements very nicely the lecture 7, and I'm sorry, it's lecture 8 on the rhizosphere, which focused on overall what's going on in the rhizosphere and on mycorrhizal fungi. So this is the important other symbiotic relationship between plants and microbes that certainly we know the most about. That along with mycorrhizal fungi. I bet there's a lot of other symbiotic relationships too that we don't know much about and we don't teach about yet. So what we're going to do is finish the nitrogen cycle, get that N2 gas back into the atmosphere, hopefully not too much nitrous oxide as well, through the process of, do you remember which process? Well, what's one of two processes where this occurs? Yeah, the main one is denitrification, nitrate being used as an electron acceptor, N2 gas being the end product, some nitrous oxide coming off, but remember also some nitrous oxide can come from the process of nitrification as well. All right, so we're going to look at overall what the process of nitrogen fixation is and the nitrogenase gene a little bit, talk about some of the types of organisms that are involved in nitrogen fixation, going from this very clingy relationship between plants and microbes, symbiotic relationships, to looser ones, to the free livers, and then focusing on rhizobia, sort of a case study looking at what goes on in the whole process, the courtship and the coming together of the two and this union that leads to this symbiosis called nitrogen fixation, and then some of the limits on the reactions. So let's go. So nitrogen fixation, not to be mixed up with other kinds of nitrogen processes in the end cycle, and sometimes people call other things nitrogen fixation. But nitrogen fixation is conversion of atmospheric N2, this triple bonded N, this very, very difficult to break bond between these two nitrogens to ammonium and then into amino acids. And diazotrophs, these trophers, eaters of this diazo, is this general term given to nitrogen fixers. So only prokaryotes naturally fix nitrogen, and the ability is fairly widespread through at least 11 of the families of bacteria known. And we used to think that it was only like a small group of organisms that fix nitrogen, but we've found increasingly more and more can. And what seems to be going on is that the genes are carried, or a number of the genes, on a plasmid, and therefore these genes are transferred through horizontal gene transfer across different unrelated groups of organisms. I do have to point out that clever little eukaryotes that we are, humans, have learned to fix nitrogen through the Haber-Bosch process, which was involved with making explosives, and under very high temperature and very high pressure, we can also fix nitrogen, that is, get this N2 form into a reduced form of nitrogen that's biologically available. But it is at a cost, so very dependent on a lot of consumption of fossil fuel, and very costly, I would say, for ultimately the environment has been very costly. The other thing that it's done is it's brought a lot more nitrogen than previously was into the biosphere. We talked about that a little bit before. So there's much more N cycling around and going to places where there previously had not been a lot of nitrogen available, and changing ecosystems and causing a lot of contamination, which is really just displaced nitrogen into a place where it can't handle that much nitrogen. Okay, so if we look at the enzyme complex responsible for N fixation, it's made up of these different components. There's a nitrogenase reductase and a dinitrogenase gene that are dependent on iron, and in this case also molybdenum, although there is some variation among these complexes that we've found more recently with other cofactors involved in these enzymes. But molybdenum is often needed, particularly for rhizobium, as a essential nutrient. And in soils where there isn't a lot of molybdenum, it can actually lead to inability to establish legumes as crops, even if they're inoculated, because there is this limitation of this absolutely essential nutrient. So what happens is that the N2 is taken up into the nitrogenase complex, which readies itself for this reaction by consuming quite a bit of ATP, which is needed to essentially break that triple bond. It takes a lot of energy to crack that thing open. So the dinitrogenase reductase is involved in that, with the transfer then of electrons over to the dinitrogenase, where then we have this N2 coming in and being converted to ammonia at this particular location. So a couple of interesting things. So, well, let's first look at the equation. So we have nitrogen gas coming in. Electrons are needed coming from the ATP to drive that, and that's shown here. These 16 ATP molecules per 1 N2 that's going to be fixed or converted then to the other side, ammonia, plus some hydrogen gas comes out. That ATP is now ADP, and you have these 16 phosphates. So very expensive, you can see that you don't do that without a lot of access to ATP and a lot of access to things like sugars to make that. A couple of interesting things here is that hydrogen gas is a byproduct of this reaction. Some organisms have ways to capture those electrons in that hydrogen gas, use the hydrogen gas as an electron donor, and be able to make energy from that to compensate for the energy used. Acetylene is a chemical that's used to estimate nitrogen fixation activity. It's very difficult to measure how much N2 has been fixed by measuring the depletion of the N2 from the atmosphere, because there's so much nitrogen in the atmosphere, and any nitrogen that's fixed makes hardly any dent at all whatsoever into that very, very high concentration. But you can put in something that's rare, like acetylene, which is used by the same enzyme complex, and you can measure this conversion and use that activity of this reaction as a surrogate or as an indicator for Nfixation activity. The other interesting thing that happens is that oxygen inhibits the reaction, because it can actually bind to the active site here and end up taking the place of the N2 gas and reducing the amount of nitrogen that will be fixed. This enzyme system is highly conserved. As I said, the genes are on plasmids. There's 21 genes altogether. They can transfer it pretty easily among each other. People have been trying for a long time to get it to transfer into plants so they directly fix nitrogen, but that's extremely hard to do, and I don't think people have been successful with it yet, or maybe not successful in a very cost-effective way. Okay. I think this is just the same equation. I'm sorry, I didn't mean to keep this in the slide. I think it's the same points that I just said. Okay, so here are the different types of biological nitrogen fixation, not including the human kind. So we have symbiotic relationships between these famous pairs of N-fixing organisms, legumes with rhizobia, actinorhizal plants with Frankia, which is an actinomycete that fixes nitrogen. We'll talk about it in a minute. And cyanobacteria are nitrogen-fixing bacteria that partner often with different groups of organisms. And we talked about that a little bit before. The free-living nitrogen fixers are ones that just duke it out alone without any access to carbon from a host or partner, and that would include cyanobacteria that live free, and azotobacter, which is what you have just made mud pies in lab, and you will be looking at them in lab on Tuesday, and because you will have enriched for azotobacter, given what you added to those little mud pies that you made, and you should see them growing on the surface. There's also associative Nfixers, which are in a more loose relationship with their host or their partner, and that could be azospirillum that are living in the rhizosphere of certain plant roots, but not in this very tight relationship where there's an actual big modification to the structure of the plant roots, like those nodules form, like there is with the rhizobia. Lichens, the relationship between cyanobacteria and fungi, another associative relationship. And there's also nodules that grow on surfaces of roots, plant leaves that are up in the air that can fix nitrogen as well. A lot of cool stuff going on. So the legume rhizobium is the most well-known. In lab, you're going to be looking at some vetch and bell bean and perhaps some clover, looking at the nodules that have come up on the roots that were pulled up for these plants that hopefully you have with you. So subfamilies of legumes, these big named subfamilies of these legumes, big words. 700 genera, over 19,000 species of legumes have this symbiotic relationship with different kinds of this general class of organisms that we call rhizobia. A lot of them have not been well studied yet. Those are the plants. The bacterial partner is a gram-negative rod. That includes different subgroups within it. And here are the different genera of organisms that fall under this umbrella of rhizobia, including rhizobium, bradyrhizobium, which I think are going to be associated with the bell bean. I'm not sure exactly. I think you're going to see both brady and rhizobia. And there's also these other ones, these cynorhizobia and azorhizobiums. And notice that they're grouped by around their host plant. And this is these are called cross-inoculation groups. So the organisms within these groups all can be associated with these particular kinds of hosts. They're bound together by the fact that they colonize the same hosts. Here's a picture of bradyrhizobia colonizing a legume, and these tend to be slower growing organisms, whereas the rhizobia are faster growing and usually have smaller nodules. Now, when you look at those nodules and do cross sections of them, or even if you don't, and if you just look at them straight on and you get the soil off of the roots, you can see this pinkish color, and that pinkish color is associated with the presence of leg hemoglobin. Very similar in structure to the hemoglobin in our body that carries oxygen in our bloodstream. This leg hemoglobin does exactly that. It binds oxygen in the nodules and reduces the availability of that oxygen by orders of magnitude, and therefore lets that nitrogen fixation process occur. And so if you see that pink color, you know there's active nitrogen fixation going on. If you don't see a pink color, then you're probably looking at a cheater. Someone who's living in that nodule, getting sugar from the plant and not doing a darn thing. And probably its game is going to be up pretty fast once the plant figures that out. Okay, so rhizobia on plates are kind of mucousy, snotty. Sometimes you can pick up a loop of colony of rhizobium, and it's like disgusting, sort of snotty consistency due to the fact that they produce a lot of extracellular polymers, polysaccharides, and this around this little rod of a rhizobium here. All this debris here is that extracellular polymer as it converts when you're preparing the cell for electron microscopy. And that extracellular polymer is really important in helping rhizobia cling to the surface of the plant root as it's growing through the soil and getting ready to like make these nodules. Okay, there's a second important symbiosis that's really common in California with actinorhizal plants and an actinomycete called Frankia. Frank, the bacterium that fixes nitrogen, not to be confused with rhizobium. So these are non-leguminous plants that are associated with bacteria that are not rhizobium. They are actinomycetes. Plants include alders, bayberries, casuarina, ceanothus, mountain mahogany. Some of these are these classic California shrubby plants, these bushes that grow around. Common more in temperate northern regions. And it's very similar to how rhizobia works. It's the hyphae in this case penetrate the root and leads to the formation of a nodule where nitrogen is fixed, and that's shown here. These are visible things to the eye. Other associations are Anabana, which is kind of a kind of cyanobacteria that associates with azolla, or like duckweed, which grows pretty profusely in any area where there's standing water. And for centuries, this azolla was promoted, the growth of this azolla and its anabanan partner, to be able to fix nitrogen and bring it into flooded rice systems in Asia. It was the main source of fertilizer long before there was any mineral fertilizer around. It was really important for growing food. In addition, tree ferns, these ancient cycads, some of which can be seen in the greenhouses at UC Davis, they also have a relationship with cyanobacteria that live in these little crevices where water accumulates. And also, of course, many lichens also fix nitrogen. Okay, so here's the infection process. I think I have to put my glasses on for this. So what happens is that when a legume is just starting to grow in a soil, it's going to put out root hairs. And with that root hair is going to be this release of an organic compound that's going to start this signaling. And they're usually flavonoid compounds that will be released into the soil and be picked up, hopefully, by a rhizobium that has a receptor for it. And then using the nod genes, it's going to take that original signal compound and add some additional pieces to it and send it back to the plant saying, yeah, I hear you. I'm interested. Look, here's my commitment, and send it back and say, I'm ready to engage in this relationship. And so at that point, then the rhizobium and the root hair are getting in close contact with each other and talking back and forth. And I don't know exactly what they're saying or how they're saying it or how even close they are to each other. But there's definitely this signaling that goes on. And then the root hair starts to curl around these rhizobia that have accumulated at this surface of this place where there's this signaling going on. And then they start to enter into the root hair. And along this infection thread here, the cells start to divide and essentially make like this thread, right, of continuous cells that starts to then penetrate the cortex cell of the plant. And so they're migrating inside the cortex of the plant, and there starts to then be a rapid division of cells in those infected plant cells infected by rhizobia that lead to kind of like the formation of a tumor, which is this nodule that then you can see actually with your eye affixed to their outgrowths of these root hairs that have been infected with the rhizobia. And this gives you a more close-up view of these cells that then start to change in their form, that they're no longer these streetwise, soil-wise rhizobia that are hanging out in the soil, living like normal organoheterotrophs. They now become these nitrogen-fixing machines, these bacteroids, these sort of mangled versions of themselves that, for the most part, most of them can no longer live freely and will not be able to live again if they are going back out into the soil at some point. But they're really good at fixing nitrogen, and they're really good at living in this this environment where they're going to be fed sugars and they will be feeding nitrogen back. And here's another picture of that, showing this curling and the rhizobia being trapped in the root hair and this infection thread growing. So it's a much more clear picture to show that particular aspect of this process. And then you can see this nodule forming on the root hair. And here's some cross sections here. It's so cool. Here's this infection thread going up inside the cell, and look at all these differentiation of the root that's bulging out this root hair and leading to the formation of this nodule. Super cool. Okay, so when that nodule is formed, then there's all different kinds of stuff going on inside it in this metabolism that is includes carbon going to the organisms that are inside these nodules, these rhizobia that are now bacteroids that are going to fix nitrogen, and the nitrogen that they fix going back to the plant. So let's first talk about the carbon metabolism. So the plant root's going to supply sugars that are fixed, often in the form of malate, that will then go and generate ATP. And there's oxygen around in this portion of the nodule to help really rapid conversion of that sugar into ATP. There's also going to be electron carriers that are going to be loaded up with electrons and as well, that will be coming out of that reaction. And then yeah, this is just repeating that those bacteroids aren't going to be very, for the most part, most of them cannot go and live free after they go into this situation. All right, then those electron carriers with those, remember with those electrons that we showed in the original reaction and that ATP being consumed are going to drive this nitrogenase reaction where we looked at the components previously in that previous slide, where then that nitrogen gas is going to be converted to ammonia. And then that ammonia is going to be converted at some point into amino acids or maybe also amides, and then go back into the plant vascular system and be translocated to where it's needed in the plant. And remember, if the rhizobia has the ability, then that hydrogen gas can also be, those hydrogens that are created can also be converted into hydrogen gas, and that will go to help fuel this reaction. Okay, oxygen, as we said before, is going to reduce or shut down the reaction. So there's two ways organisms involved in this. These bacteroids help keep the oxygen away. One is physically protecting the actual reaction site for the Nfixation through a diffusion barrier where it can't get in. And then the other is that pink color of that leg hemoglobin. That leg hemoglobin is going to carry keep the oxygen from being available inside the nodule until it's needed, for example, here at the generation of ATP. Okay, so limits on Nfixation in general, and you can sort of think about what's the advantage of being a symbiotic relationship, being in a symbiotic relationship versus being free. Even though being free is great, freedom is wonderful, but here are some of the trade-offs. So very expensive, you need 20 to 120 grams of carbon to fix 1 gram of nitrogen, so that's a lot. So if you have someone who's providing you with that carbon, then you're going to have a much better shot of being able to afford fixing all that nitrogen and being able to, have a good life. Other sources of nitrogen fertility will strongly inhibit this nitrogen fixation. So if the plant has access to a lot of ammonium and possibly nitrate as well, then it's already going to have the end product of the Nfixation, right? Because the nitrogen is converted to ammonia. So it's going to say, I don't need to be paying all this sugar for this process. So it'll shut down Nfixation in nodules. And then we have oxygen. So different strategies, different groups of organisms use, because remember, these are limits on Nfixation in general, is the physical protection in the nodules where there's actually a diffusion barrier, but also in cyanobacteria, they actually have these heterocysts, which are these specialized cells that are very thick-walled, where nitrogen fixation goes on in them in this filamentous organism, where the other cells in the filament are specializing in making ATP. Also, there's avoidance in some systems, especially if there's stratification in the environment, like in a flooded soil, may go down to a deeper level in the absence where there is no oxygen and fix nitrogen during that time that they're down there, and then maybe move back up to make ATP. Some organisms live microaerophilically, and can fix a little bit of nitrogen, but at the cost of not making as much ATP. And there's other organisms that pant. They produce a whole lot of CO2 almost in a wasteful way to just convert that oxygen. So they're using the oxygen as an electron acceptor, but sort of at the tail end of the process and not gaining energy from the reaction, but essentially just sort of getting rid of that oxygen by converting it to CO2, and I just call that panting. All right, estimated rates of Nfixation. The highest rates are for sure associated with the symbiotic relationship. So here are these leguminous plants. Their rates of nitrogen fixations are in the 100 and above of kilograms of nitrogen, dinitrogen fix per hectare per year, whereas free-living bacteria like that azotobacter in your mud pie can be quite small, can be very, very, very minimal compared to what we see in the symbiotic relationships. But it may be all that's there, and maybe it's in ecosystems where there isn't that much productivity and not that much need for nitrogen. So it can be very important, like in tropical ecosystems. And actually, I think that there's new research coming out showing that the free-living nitrogen fixers are much more important than we used to think. Lichens aren't too shabby. They can fix rates that are on the order of half of the legume relationships. And then the Frankia actinorhizal relationships are also pretty high. Not quite as high as the legumes, but up there. But you can see it really does pay off in terms of how much nitrogen can be fixed to have a symbiotic relationship. And of course, we really tap into these in agriculture using these plants and their associated bacteria to get a lot of nitrogen into systems, like you saw at Russell Ranch in those cover crop systems and the organic system. All right, that's it. There's a little bit of supplemental information that talks a little bit about survival of rhizobia outside the nodule, and just a little how-to, a cartoon for how to inoculate your seeds with an inoculant of rhizobia, which of course you can go to Ace Hardware and buy it, or you can buy seeds that already are coated with the inoculum. Do you need to add the inoculum? What an interesting question. Well, I think it's quite beneficial when you first start to plant legumes in soil that hasn't had legumes for a long time. This is what people say, at least. Then it's nice to have that inoculum to jumpstart it and get it going. But after a couple of years, there's no benefit to keep adding inoculants. And in fact, the organisms that will be those responsible for nitrogen fixation, which you can look at by taking those nodules and isolating DNA from them and seeing, okay, who's in there? Is it the organism that was in the original inoculum? And you might find, nope, no way. It's organisms that are living freely in soil that maybe were present at low numbers at in the first place, but now have increased in their densities. Okay, and then some nice links, fact sheets for you, and that's it, and good night.